if someone came to you and said, okay, I've talked my, let's say, family member into not doing a certain cancer treatment that will, completely mess them up, but I don't know what the alternative is. What advice would you give them? Because I believe that, being armed with this knowledge will be very helpful in a situation where a family member might, get cancer.
Yeah, well, that's a great question. And I have to say that I know you said deuterium is coming later, but I'm going to bring it up because I believe that deuterium is heavy hydrogen, and deuterium toxicity, I think, is a driver behind cancer. I actually almost believe that it is the cancer cells are able to sense deuterium levels in their environment, and when deuterium is high, they kick in and they start reproducing and making more cancer cells. And the reason why they do this is because they know how to turn off their mitochondria, and the mitochondria are very sensitive to deuterium. It makes them, it causes the mitochondria to become inefficient in making energy, ATP, and also releasing all kinds of reactive oxygen that damages the cell. So the cancer cells are, their job almost, in my opinion, is to sense deuterium levels, and when they get too high, to start proliferating. And they turn off their mitochondria. They use their mitochondria to generate the nutrients that go into proliferating. And instead, they take in massive amounts of glucose, sugar, and convert it into lactate. And then they release the lactate into the circulation and supply it to the rest of the body. The brain loves lactate. Lactate allows the brain to very efficiently make ATP without having to go through glycolysis. So the lactate is supplying the deuterium depleted nutrient to the entire organism, while the cancer is busy proliferating to make more cancer cells so they can make more lactate. And the system is designed to try to protect the mitochondria systemically from deuterium damage, which is essential because if the mitochondria in the brain can't function, you get things like Alzheimer's and Parkinson's disease, all these nasty neurodegenerative diseases that I think in many ways are worse than cancer. So I don't believe in killing the cancer. I think the way you treat the cancer is to nourish it. And that involves eating low deuterium nutrients, getting rid of things like glyphosate, the toxic chemicals in the environment, eating organic foods, getting lots of sunlight exposure, sort of basic healthy living. Convert to basic healthy living as much as you can and eat very nutrient dense foods. And then, so you're basically working towards healing the whole system so that it can manage the deuterium toxicity and get the deuterium in the right places, because the body has very fancy mechanisms to pack deuterium into the bones and into the fascia, and then to keep it away from the mitochondria, and also has very efficient mechanisms to deliver low deuterium water to the mitochondria. These systems are really elegant and they're basic to all of metabolism, and glyphosate derails them in many, many ways.
Wow.
So that's a
So
huge introduction to much more than you expected, I'm sure. But I definitely do not think you should take any of those. Chemotherapy is not only, of course, killing the cancer cells, but also killing a lot of your immune cells. I mean, it really causes so many side effects that are really much worse than the cancer in many cases.
so practically speaking, let the cancer run its course.
Exactly. And once
Wow.
the cancer is able to heal the immune cells actually swarm into the cancer tissues, but they don't attack the cancer. The cancer tells them to stand down. And in fact, they're happy to stand down because they're getting all these wonderful nutrients from the cancer, which is helping them to heal themselves and their own mitochondria so that they can actually clear the cancer. So you want to, it's really counterintuitive. You want to nourish the cancer so that it can support the immune cells to fix them so that they can kill the cancer. Let the immune cells kill the cancer by fixing them. But instead, what we do is we attack the immune cells with the same chemicals that are attacking the cancer, which makes everything worse. And then the cancer gets metastatic. Metastasis is what really ends up killing you with cancer. And the chemicals increase the risk of metastasis.
Wow. I remember listening to, your conversation with Joey, because you did an episode with
Yeah,
him.
I love Joey. He was so fun. Yeah, I really remember him so fondly, and I appreciate that he still got my interview up there. So
And I remember you saying that saturated fat is one of the healthiest things you can eat. I believe it's because it creates deuterium depleted water, right?
that is right.
Could you
Yes,
could you talk more about saturated fat? Because generally
yes.
speaking, it's pretty vilified.
I know. That's also I have to say, pharmaceutical industry has got everything just about backwards. It's almost the case that if they're recommending something, run away. Don't do it. Just take their advice and negate it is really the way to stay healthy, in my opinion. I can't believe how many ways they've got things wrong. It's just really shocking. But the fatty acids, so that the saturated fats in particular, are healthier than unsaturated fats. A lot of people are taking these pills of PUFA, polyunsaturated fatty acids. That's not good because those are useful to you, but only because they are metabolic products of reduction of the fat. You start with the fully saturated fat, and your body turns it into the unsaturated fats. And in doing so, it produces deuterium depleted water. So when you leave out that step of starting with the saturated fats, instead you go straight to the unsaturated, you're missing the whole point of what's healthy about those unsaturated fats. They work well in certain places in the membrane that the brain has learned how to use them appropriately, but the body wants to make them. It doesn't want to receive them in the diet. It wants to make them from the saturated fat, because in making them, it can release all these hydrogens that are in the saturated fat that are low in deuterium. And so the whole goal is to make deuterium depleted water to support the medium of the mitochondria, specifically in the mitochondria, which are the energy. These are little organelles inside all the cells, and the mitochondria are able to make the ATP, which is the energy for the cell. But those ATPase pumps get destroyed by deuterium, and the whole metabolic system is aware of that. It's constantly aware of the fact that we got to get low deuterium water into those mitochondria. And it's so fascinating to go back and look at I have been studying metabolism for a long time. I'm fascinated with biology, and I happen to have a lot of knowledge. And once I realized the deuterium problem, then everything made sense. It was so cool. I was so excited because all these things that are, and even these, molecules that are so special, like melatonin, and even cholesterol. Cholesterol is actually a low deuterium nutrient.
So, with that said, how do we avoid deuterium?
Yes, well, so start by eating highly saturated fats and cholesterol, and eating organic, of course, because the thing is that the microbes in the gut are central to getting started on the whole process of lowering the deuterium levels in the mitochondria, the microbes in the gut. And glyphosate critically hurts certain normally abundant microbes in the gut that start the whole system that produces very low deuterium short chain fatty acids. This is acetate, butyrate, and propionate. They're very little, Then you have these long fatty acids, and then you have these little stubby guys. But the short chain fatty acids come directly from extremely deuterium depleted sources because of a really clever trick that the microbes do. And this is really fun. This is where I sort of began to get really interested in deuterium, when I realized that the microbes, they have an enzyme called hydrogenase, and hydrogenase can take hydrogen atoms off of organic molecules like glucose. They can take the hydrogen off, put two hydrogens together, and make hydrogen gas. And our cells can't do that. And you have all these gases, people get bloated and everything. That's because they've got these gases that are not going back to organic matter, because glyphosate's messing up the enzymes that bring them back. And these are microbial enzymes. So the microbes make the hydrogen gas, and they use the hydrogen gas to turn carbon dioxide into methane or into acetate. We have microbes in our gut that can do both of those, methane and acetate, from carbon dioxide, using hydrogen as a reducing agent, hydrogen gas. And why that's important is because that hydrogen gas has lost 80% of its deuterium. And that's because deuterium is heavier, right? It's heavy hydrogen. It has an extra neutron, which makes it twice as heavy as hydrogen. It's natural. It's all over the place in nature. You can't avoid it. It's a natural element. But the biology has to deal with it because it goes everywhere hydrogen goes. And if you have a glucose molecule that happens to have a deuterium there instead of hydrogen, the enzyme that makes the hydrogen gas will ignore that deuterium. It'll never take it, almost never, take that deuterium and turn it into a hydrogen that has a deuterium in it. HD, for example. It'll almost never do that. That's why you lose 80% of the deuterium when you make the hydrogen gas. And now you take the hydrogen gas and add it to carbon dioxide. Now you've got, organic molecules with carbon-hydrogen bonds in the methane. Methane is CH4. So it's got two hydrogen molecules combined with the carbon of carbon dioxide. And then it releases oxygen. The oxygen part of it goes out and does other things, but the CH4 is an extremely low deuterium nutrient if it came from that step by the microbes of reducing carbon dioxide to methane. And the same thing for acetate. Acetate is basically a methyl plus a COOH. So it needs another carbon dioxide to stick the COOH onto. This is getting a little bit of chemistry, but acetate's not too far away from methane. And once you have acetate, then they can make the other two the butyrate, is two acetates basically make butyrate. The microbes do that too. The microbes make all of this, the acetate, the propionate, and the butyrate. I think our cells don't even know how to make the butyrate. We're depending on the microbes enormously for these nutrients that are being produced in the gut from carbon dioxide and from reduced hydrogen gas that's lost 80% of its deuterium. So those short chain fatty acids that the microbes produce are extremely nutritious, very low in deuterium. And the colonocytes lining the colon love a butyrate. That's their favorite food. And if they don't have enough butyrate, they're not healthy. And that's, I think, because the butyrate is so low in deuterium.
Yeah, that's something I realized a little while ago, that bacteria are actually the ones that supply nutrients to the gut lining. So if you have a problem with the gut lining, then you need bacteria. It's not just
That's right.
about, supplying the nutrients to the gut lining. It's the bacteria that produce certain nutrients that the gut lining needs. But
Exactly.
speaking of you know, keeping your bacteria alive, one of the things that glyphosate does is it kills bacteria. And in your book, Toxic Legacy, you
Mm-hmm.
make the case that glyphosate is the most dangerous thing we're being exposed to today from a pollution perspective. Now, why is that?
Yeah, I think it's the most dangerous because number one, it's the most common. It's the most commonly, I think it's the most commonly found toxin. When you look at food, you take a sample of, say, fast food, and you analyze, for the different toxic metals and the insecticides and the fungicides and some antibiotics. You find all that stuff in there, but what you find dominantly is glyphosate. 95%. So Zen Honeycutt's a friend of mine, and she's done a lot of different types of food categories looking for glyphosate. And she did school lunches at the public school system in the United States. She found 95% of the school lunches tested positive, of the samples tested positive for glyphosate. She did fast food, like McDonald's and all those fast food restaurants. 100% of the samples tested positive for glyphosate. So when you look at something else, a fungicide or something, it's a much smaller percentage of the foods that actually have it. Glyphosate is everywhere in our food supply because we extensively use it in agriculture to make food. And the farmers think they can't live without it. And of course, the government thinks it's completely safe. This is the message that they got from the chemical industry. The industry has fooled the government into believing that it's perfectly safe for human consumption, which is, I think, a huge lie. And I can't believe that the industry itself doesn't know that it's extremely toxic. And as I said to you before we started, there's a really unique diabolical mechanism of toxicity that glyphosate has. That I believe glyphosate has. It's still, you could call it theoretical, but I think the evidence in my book is very compelling, I believe, to convince people that that's true, including from Monsanto's own studies, really showed this loud and clear, that glyphosate it has a unique mechanism which involves substituting for the amino acid glycine during protein synthesis by mistake. So everywhere a glycine could be, a glyphosate could slip in instead by mistake because it is a glycine molecule. It's a complete glycine molecule, except that it has extra material stuck onto its nitrogen atom. And that extra material is a huge problem because once you put that into the protein is depending upon glycine to be very small. Often at very critical sites, the protein depends on that. That's why glycine is there. And when you put the extra stuff from the nitrogen atom into the space, all of a sudden the substrate doesn't fit anymore and the enzyme is dead in the water. So it kills these enzymes when it does. Many different enzymes are very sensitive, very susceptible to extreme disruption by a single substitution of glyphosate for one glycine in that protein. It's really fascinating. And you can actually go hunt down all the proteins. And I've done that for many years. I still find new proteins new to me that oh my God, this one's going to be susceptible for sure. And then, oh my God, this has terrible consequences. I just found one recently, which was a dehydrogenase that actually reduces saturated fats, and turns them into less saturated fats. A dehydrogenase that is critical for fat metabolism. And if you have a deficiency in that enzyme, if you have a genetic defect in that enzyme, you end up as a child, you end up with lots of problems, including autism and also picky eater. You really don't like a lot of foods, and we have so many kids in our country who are very sensitive to different foods and refuse to eat anything but sugar. We have a huge problem with the kids in this country right now being picky eaters. And this if that enzyme is disrupted, you will be a picky eater, and also you'll have more susceptibility to autism.
One thing I found really interesting is when you were talking about the specific type of bacteria that actually detoxifies glyphosate, and not in the soil because there are a few in the soil, I believe, but in
Hmm.
the digestive tract. And I don't think
Yes.
it was just a bacteria. There was also, I believe, Candida also does it, right?
Yes, you're right.
Yes.
Well, let's see. I think Candida. There's also another fungus that does aspergillus.
Yeah.
Does a good job. In fact, they're even proposing to use aspergillus as a way to clear glyphosate from the soil, and that, of course, is a fungus that causes trouble. I mean, it's a and there's Pseudomonas aeruginosa is another one, which is a big problem in the hospitals, causing nasty infections. But it can clear glyphosate. I think that there's only a few organisms that can actually break down glyphosate because it has this really tricky CP bond, and most of the bacteria are stumped by it. That's why they just get killed when they're exposed to it. But the critical ones that survive do a service for the host because they remove the glyphosate by breaking it down. Then you've got the phosphorus and nitrogen you can use as nutrients, you know. So Pseudomonas, if it grows in your gut, you're probably not going to have a problem with glyphosate, but you're going to have a problem with Pseudomonas. And so then when you take an antibiotic, I actually am fascinated by, see if I can get this right, Cipro. So Cipro is a class of, is in a class of antibiotics that people are given quite frequently to try to control a Pseudomonas infection. And Cipro has really unusual side effects that happen months later. It's really, I've been reading a lot about it because I'm so fascinated. Cipro kills the Pseudomonas, right? So you've got a Pseudomonas infection, you take Cipro, it kills the Pseudomonas. And then 6 months later, you start having problems with your tendons. You know, tendonitis is a side effect of Cipro that nobody can figure out how it causes that. But you could just take one round of Cipro to cure an infection. And 6 months later, you've got tendonitis. Well, that's because you killed off the Pseudomonas and now you're being poisoned by glyphosate. That's what I think. Now, this is a theory that I haven't actually advertised very far. We might be the first to hear it, but I am suspecting that could explain why this class of antibiotics is causing these really bizarre after effects and often long after you took the drug. So it's very mysterious as to how that happens. And I think this is an explanation.
There's a guy called Argenis Vonderplants, and I was thinking of this man when I was reading that specific passage. Um, so his basic theory when it comes to microorganisms in the body is that E. coli and Salmonella and Campylobacter and all of these dangerous pathogenic bacteria are actually, detoxifiers. They're a natural part of the body. And if you have symptoms, it's because it's in conjunction with pollution.
Mm-hmm.
It's the same with candida. It's the same with any fungus. He says that if they're present and growing in the body, that's because they're performing a beneficial function.
I love it. I love it. That's exactly what I say too. I actually see that with the fungus and also, of course, with the cancer, as I mentioned earlier. All of these pathologies that we're experiencing, underneath there's a goal. The organism as a whole is actually taking advantage of something essential that particular pathology, the source from those organisms is achieving something really important for the host. And the sort of the whole system has decided in a way that that's a better alternative than to let that thing happen, which and you can see with the Pseudomonas, if you kill them off, now you're going to have glyphosate. It's protecting you from glyphosate toxicity. And at the same time, it's giving you symptoms that are uncomfortable. So I think that's absolutely true. And actually, Candida is very interesting with respect to cancer. And I just actually wrote a paper, which I'm hoping I can get published. It's not yet out, and in that paper, I talk about cancer and what I just told you earlier about cancer being protective of the mitochondria with the deuterium problem. Candida does that too. And it's actually very analogous to cancer because when you have a massive yeast overgrowth, the yeast cells love sugar, right? Just like the cancer cells, they're very similar. They love sugar. And when they process the sugar, they release ethanol, actually alcohol, not lactate, but ethanol. But it's a very similar pathway, a little bit difference between ethanol and lactate, but not much. Ethanol is also an alternative source of nutrients that can feed into the mitochondria of the entire body. And so the candida, when they overgrow, they release ethanol, and you can actually get drunk without drinking any alcohol by eating a lot of sugar and having a lot of yeast. You can get very drunk from that. And I think a lot of the brain fog may be related to that, just having too much ethanol in your brain produced by the yeast that are flourishing in your gut because they're helping to supply the ethanol that they supply is going to be a low deuterium nutrient, which makes me think that organic wine is actually a good food source,
Yeah, that's very interesting. Now, the last thing about glyphosate is that I even if you buy organic, you're going to find it in the environment as long as it's used on a large scale in a country. You're going to be
Yes,
exposed to it, whether you want that or not. So
right.
when I'm sat and thought for myself, how to avoid glyphosate, I thought, okay, well, the only option is to move to a country that doesn't
Yes.
use a lot of glyphosate. So maybe I'm not sure, but maybe the reason you're in Hawaii is because you're trying to avoid glyphosate. And
Absolutely. In fact, I live on the North Shore of Kauai where there's a lot of organic farmers and we have a lot of good organic food that we can buy from the farmers. And that's certainly why I'm here, is to get away from places where you're going to be exposed. And I have to say, even I think in the cities, you're being exposed to glyphosate that I believe is in the biofuels. This is another thing I'm worried about. We've been really ramping up the biofuel industry where we grow corn, it's GMO Roundup Ready corn, spray it with glyphosate, turn the corn into ethanol, put the ethanol into the gasoline. And now when you're at the gas station filling up the tank and it's hot and things are evaporating, the glyphosate's going to get into the air and you're going to breathe it. So I think the cities, and in fact, I have my friend Anthony Samsel, who's a toxicologist. We collaborated on a number of papers, and for a period of time, he was collecting water samples from his friends to send to him and get tested for glyphosate. And I gathered from rain. So I got a sample from rain in Cambridge at MIT, where I work, a sample from my home in Winchester, which is a northern suburb of Boston, and a sample from here in Kauai. And the two suburban samples, the Hawaii sample and the Winchester sample, were negative. They didn't have glyphosate, but the one from Cambridge at my office did. It tested positive for glyphosate in the middle of the city where you don't have a lot of farmers growing, glyphosate contaminated crops. But what you do have is a lot of trucks using biodiesel, and you have a lot of cars using bioethanol. And I think that that's causing the air in the city to be very toxic. So I really worry about that. In fact, kids who live near highways have an increased risk to autism. I suspect that's because they're picking up glyphosate when they breathe the air.
wow, so glyphosate is that stable? I'd think that, because of
It's hard to break down.
my God, okay. Well, we really need bacteria then.
I
Well,
know, I know. Yeah, a few bacteria that can do it. Now, sunlight will also kill glyphosate and also say chlorine. I was really happy when I learned that chlorine can break down glyphosate non-enzymatically because we typically use chlorine to purify the water. We're trying to kill off of microbes, in the drinking water supply, in the municipal water supply for the different cities. They put chlorine in that water to kill off the microbes and make it safe to drink. But they're also killing off the glyphosate when they do that. And I was like, thank God, because otherwise you've got all this water washing down from the farms, and getting into that. It's just really dangerous. If we didn't have that, I think we'd be much worse off just from the drinking water if we didn't purify it with chlorine.
so Hawaii seems to be one place that you can go to avoid glyphosate. Do you
Mm-hmm.
know of any other places in the world?
Bhutan.
Tan, okay.
Bhutan. I think there. I even was considering moving there for a while. My husband, no, no, we won't adjust. It's too the culture is too different. So yeah, we've been looking for places ourselves. And Bhutan, I think, is probably, although it's changing because they're getting more and more westernized, but they're basically small family farms. That's what you're looking for: small family farms. Lots of Africa is also probably much less glyphosate, again, small family farms. But the industry is trying madly to get all these countries to convert to the Western style agriculture, which reduces labor considerably. Really, we have, the United States, probably half the people used to be farmers back before we had these kinds of industrialized agriculture with these huge farms and very little need for humans because you just poison everything so that way you can grow your crops very efficiently. And we pay a huge price in the healthcare costs, and of course in just the general well-being of the nation. I mean, all these autistic kids, we're really struggling, the U.S. with all the healthcare problems that we're facing. We have a huge, much higher healthcare expenses than any other country in the world. We spend the most on healthcare. We don't see a return for that for our efforts because we have a very low life expectancy compared to other industrialized countries, very high infant mortality. I mean, we're doing really badly on all the metrics, despite all this money that's being spent on healthcare. So we're doing something very, very wrong in this country. And the government doesn't even seem to be concerned about it, which really frustrates me. It frustrates me that this isn't a topic of conversation around the election. Which president? Who do you want for the president? Well, I want to know how they feel about the farming, in this country. Do they want to do anything about it? And neither one of them seems to have any interest in even talking about that.
Yeah, we got to vote for what's he called?
Robert F.
Robert F.
Kennedy
Kennedy. Yeah,
Jr. He's
yeah,
my hero. I would love it if he could win. It would be such an upset. I would just love it. I think he's who we need as a president, and we'll see.
yeah. He actually cares about health. He actually talks about it. He's
Exactly.
the only one.
Yeah, that's his main focus, I think, is the health, and also his vice presidential candidate, uh, Nicole, I forgot
Nicole
her last
something.
name. But yeah, she's also very much into regenerative agriculture, which is like I think the number one thing we should be worried about right now, changing the farming methods in this country into regenerative agriculture, not just organic but regenerative. We need to fix the soil. The soil is a mess because glyphosate's been destroying all the microbes in the soil, depleting the nutrients, the micronutrients like the minerals. I mean, we have really bad soil, and then we have really bad food because it's depleted in nutrients because the soil is getting wrecked. I mean, it's just all the way down the chain.
Speaking of making the public healthier, there was this part, in the last chapter of your book about sunlight, which was really interesting. I've heard some of that stuff from Jack Kruse, but this, exclusion zone and making the mitochondria more efficient and red light, all of these benefits that people don't know about at all regarding sunlight. I mean,
I'm glad you brought that up. Yes. Do you want
yeah,
to go
I
ahead
mean,
or?
could you talk more about that? Because I find that really interesting.
Absolutely.
Most
And
most
actually, I've
yeah,
go
most
ahead.
people always
Yeah, avoid
associate
the sun.
no, yeah, that too, but they always link sunlight with vitamin D, but there's so much more there.
Absolutely, yes. And in fact, I'm very excited about sunlight. And I started getting, I mean, I've been interested in sunlight all my life, actually. I've always worshiped the sun. I never used sunscreen, and I feel that it's a reason why I'm so healthy at the age that I am now. I don't have any health issues at all. I don't take any pharmaceutical drugs. And I'm 76 years old, so I should have some problems, at this point. But so far, luckily, I haven't. But sunlight, I think, is a critical part of that. And even sunlight exposure to the eyes. I never wore sunglasses, and I don't have any problems with my eyes. I actually get by without glasses. I don't have 20/20 vision, but I don't have to use glasses to get by. Regular glasses, my vision is good enough to get by. Anyway, sunlight. So it's really fascinating. And I got interested in Gerald Pollack's work even before I knew about glyphosate. I was really fascinated by his work on exclusion zone water. And he's a good friend of mine at this point. And he found that sulfate is very important in the glycocalyx. The glycocalyx is this whole the stuff that lines all the blood vessels is called the glycocalyx. And it has some proteins, and then attached to those proteins are these things called heparan sulfate proteoglycans. They have kind of a complicated name. But so sulfate, heparan sulfate, the sulfate, there's a lot of sulfate in those heparan. So they're actually complex modified sugar molecules all hooked together with sulfate sticking around in various places throughout the lining of all the blood vessels. And those sulfates actually have the effect of gelling the water. They make jello. So it's familiar to you when you buy you buy the gelatin, right? And you put it's powder, you put it in the water, boiling water, you let it sit in the refrigerator, and it gets very hard. There's only a tiny bit of that powder, and then the water turns into gel. And that's actually a good model for what's going on in the glycocalyx. The gelled water is also called the exclusion zone water. And I think that's a term that was coined by Gerald Pollack. And it's really, really fascinating because he showed experimentally that what happens in that gelled water is that it actually makes crystals out of it. It's kind of like, I call it liquid ice. So it puts the water into organized structures, and it pushes protons out. So it becomes negatively charged just naturally through biophysical mechanisms. It pushes protons out, and the protons go out into the blood. And so it becomes negatively charged, and it creates a battery. And that battery is then a source of energy. And in fact, when you're exposed to red light, he showed it experimentally that exclusion zone, that gelled water grows by a factor of 4 in exposure to red light. So the light, somehow the photons are triggering some reaction in the gel that magically makes the gel grow bigger. And this is a really important feature of sunlight, to make the gel grow bigger, because then those batteries get stronger. You've got solar panels, you could say, in your blood, and they're able to efficiently use the energy in the sunlight to produce the battery, and then also to push out the protons. And those protons are not going to be deuterons. They're going to be much less likely to be deuterium, I believe, because deuterium sticks better it sticks better to the oxygen than the water molecules. It doesn't let go nearly as easily as the protons do. So it not only does it deliver protons to the blood, but it also delivers low deuterium protons, just like the hydrogen gas, low deuterium protons. So the blood becomes reduced in deuterium, and then the gel becomes increased in deuterium. So the body ends up trapping the deuterium in the gel. And this is what goes on in the bones, actually. The body traps deuterium in the bones. And there was a fascinating paper that I should mention Laszlo Boros, because he's the one who introduced me to deuterium in December of 2019. He basically he sent me email, blind email, and he said, complimented me on a paper I just published. And he said, basically, by the way, do you know about deuterium? And I didn't, but then as soon as, and he had a paper that he attached that he had written, which was so fascinating. And that just launched me on this whole obsession with deuterium from that point on. And so the deuterium gets trapped in the bones. And actually, there was a study on seals, if you can believe this, on seals, where they found that the seals had twice as much deuterium in their bones as the amount that's in seawater, sort of the normal amount that you would expect if everything was just mixing up, twice as much. So the seals were able to concentrate deuterium in their bones. And the article said that they did that in order to make the bones strong. So to protect the bones from the high crushing pressure that they get when they dive deep into the water. So the seals needed to have a lot of deuterium doping in their bones to keep their bones strong. So my conclusion is we do that too. Maybe not as well as the seals, but we do that too. We put deuterium into the bones. But the mechanisms that do that depend upon things like collagen, which collagen has tons and tons of glycines, and it gets messed up by glyphosate. So I think people in America have all kinds of problems with their collagen and not forming the crystal properly, and just probably not even being able to dope the deuterium as well as it should, such that we don't efficiently push the deuterium into those places where it's safe, like the bones and the joints, and probably even the hair. I mean, you sort of put the deuterium everywhere in the skin. There's a lot of, it's just, it's become fascinating to me to think about this idea of concentrating the deuterium in the places where it's not going to cause harm, in fact, where it's going to cause good, making the bone stronger. And then that way, keeping the deuterium out of the mitochondria.
Exactly. One of the things you say in the book is that I'm not sure you say this, but could one make a case for the fact that sunscreen is the thing that's causing skin cancer much more
Yes.
than, let's say, excess UV?
Yes, I think so. And I did write about that in the book. And that's something that I, again, was aware of even before I knew about glyphosate. I was really concerned about sunscreen. As I said, I never used it. It has toxic chemicals in it. It actually has aluminum in particular. It's used as an emulsifier to make it sort of, so it's not white, it becomes kind of invisible and it spreads more easily. They use aluminum, and aluminum gets absorbed through the skin, and aluminum messes up an enzyme called eNOS, endothelial nitric oxide synthase, which I've studied deeply and which I believe actually makes sulfate as well as nitrate. Normally it makes nitric oxide, which oxidizes to nitrate. I believe it also makes sulfur dioxide, which oxidizes to sulfate. And it does this when it's in the membrane of the cell. It's really a fascinating molecule because it can attach to the membrane or it can go into the cytoplasm. And when it attaches to the membrane, it doesn't make nitric oxide, but it does. But I think it makes something else instead, which is sulfur dioxide, which becomes sulfate. And in fact, the red blood cells, there's lots and lots. They're the most common cell in our body, I believe, cell type. And the red blood cells actually have eNOS. They have that they produce that enzyme. And it's a puzzle because eNOS makes nitric oxide, which actually acts like carbon monoxide and prevents the red blood cell from holding oxygen. So it's toxic, basically, like carbon monoxide. So there's a real puzzle as to why do the red blood cells have eNOS, because they certainly don't want to make nitric oxide, but their eNOS is all attached to the membrane. It doesn't go inside the cell. And in the membrane, it can make sulfur dioxide, it can make sulfate, it can populate the red blood cells with sulfate on their surface, which will make them negatively charged because it'll build the gel around the red blood cell, make them repel each other, keep them separated, and everything's good as far as the circulation is concerned. So I think that's really a fascinating idea. And sunlight stimulates eNOS to make sulfate, which is probably why it grows the exclusion zone water, because the sulfate's needed to make the gel.
Hmm. So, what do you think is causing the skin cancer? Is it just the sunscreen? Do you think it's the only factor, or do you think glyphosate? might be involved? Do you think UV might be causing a certain chemical reaction with something that's in the skin that might be, you know, there that shouldn't be there, and so on and so forth?
Absolutely. And in fact, melanin is the skin tanning agent, melanin in the skin. And melanin, it comes from, melanin comes from tryptophan, tyrosine, sorry, it comes from tyrosine, which is one of the 3 aromatic amino acids that come out of the shikimate pathway, which glyphosate famously disrupts. So glyphosate messes up the shikimate pathway in the gut microbes, which prevents them from making enough tyrosine for the host. And with inadequate tyrosine, you get inadequate melanin in the skin. And melanin is the natural sun protecting agent that's absolutely essential for mopping up the radicals that are produced by the photons from the sun. The high energy, high frequency photons of the sun produce these free radicals from the oxygen in the skin that can cause a lot of damage. But the melanin is able to soak those up, and the melanin, of course, also makes you brown, makes you darker, so that you don't you're more protected from the when you get your tan, you're much more protected from the sun's exposure. And without the melanin, you're not going to get tan, you just stay white. And then you continue to be susceptible to sunlight poisoning, which it can cause cancer. But the other thing I always think about with any kind of cancer is, well, that's going to help the deuterium problem. So even perhaps the skin cancer is increasing systemically because of the fact that we have this deuterium problem. And anywhere that a cancer can grow, it can help to supply the body with the low deuterium lactate that can solve the problem, help to solve the problem.
Hmm. Really interesting. So that might be why some people are in the sun and then, they don't get darker because they can't produce melanin. And
That's right. And I think
maybe that
that's a consequence of glyphosate poisoning.
yeah, I was going to connect it to that the fact that
Yeah,
glyphosate kills the bacteria that produce those amino acids, right?
exactly.
So we can
Exactly.
produce
Yes.
melanin. Very interesting, very interesting.
Yeah.
Something I read in your book, which I found really interesting, is that there were these genetically modified mice, and they genetically modified them in the way where they started producing PEPCK in the liver
Right, right, right.
tenfold. And I believe you said
In the
that,
muscles, that was it in the muscles? I'm a little rusty, but PEPCK is definitely produced in the liver. A lot of it, yes.
yeah. So they started producing more of this because of a change in the genes, and they started eating more and they had more energy. But the thing that was the most surprising is they lived longer.
Yes.
So then my question is, how do you increase it?
Yeah, well, PEPCK is a very interesting enzyme. It's one of the ones that early on I detected as being susceptible to glyphosate toxicity. It actually has a very good model that's very similar to what happens with EPSP synthase. That's the enzyme in the shikimate pathway that glyphosate famously disrupts. Nobody argues about that. It suppresses EPSP synthase. And when you look at exactly how it does that, to me, it becomes very clear that it does it by substituting for glycine, because EPSP synthase has a highly conserved glycine residue. That means that the glycine is somehow essential for the protein at the place where it binds phosphate. And so I've developed this concept of a glyphosate susceptibility motif, which involves having any protein that has a glycine that's highly conserved, meaning that it shows up in all the different versions of that protein that show up in different species. It's something important about that glycine. And that glycine binds phosphate in some substrate, which a lot of proteins do that because phosphate is very important in all these energetic reactions. Then that protein is going to be highly susceptible to glyphosate, because glyphosate can substitute for that glycine. Phosphate needs room, so the glycine is there at the phosphate binding site for good purpose, which is to provide room for the phosphate of the substrate. But then glyphosate puts its methylphosphonate unit into the place where the phosphate was supposed to go, and now the phosphate can't get close, and the enzyme is dead in the water. It can't do its job. And I think that's exactly what happens with EPSP synthase. And the industry has done a lot of research on EPSP synthase and has shown that if you replace that glycine in the protein with alanine, which is a minimal change, it has an extra methyl group, which is much smaller than the methylphosphonate that glyphosate has. If you replace it with that, it hurts the enzyme. It doesn't work quite as well, but it still works. But it completely destroys glyphosate's ability to do anything to harm it. And so that's because you put the wrong code. When you put a different amino acid into the protein, it no longer is glycine, and therefore it no longer glyphosate no longer fits. So it doesn't disturb it at all. It can't substitute for alanine. It can only substitute for glycine. So by changing the code for the amino acid to be alanine instead of glycine, you solve the problem of glyphosate substituting. So it turns out PEPCK binds the same molecule, which is PEP, phosphoenolpyruvate. That's the molecule that EPSP synthase binds at the place where it binds phosphate. And it also has a critical glycine residue, highly conserved, at the place where it binds PEP. So it's binding the same molecule that EPSP synthase binds. It has a highly conserved glycine there. And therefore, I would predict that it's highly sensitive to glyphosate substitution, which will destroy its ability to do its job. And PEPCK is a very, very important molecule in the enzyme in the liver. And it really kicks in when the child is born. And it has to do with actually producing sugar. And it's very important because it can produce sugar. It's called neogenesis, I suppose. I forget the word of gluconeogenesis. Got it. Gluconeogenesis with the PEPCK. And so when the blood sugar gets low, the liver kicks in and produces sugar and puts it out, right, to raise the blood sugar in the blood. When the newborn is born, it actually needs to get PEPCK going very fast because the blood sugar can get very low in between feedings. And the infant can go into a coma if it gets too low. And so when the infant, at night, for example, when they're sleeping, if they go for many hours without having nursing, they can get very low blood sugar, go into a coma, and you can get sudden infant death. So I suspect that glyphosate is a causal factor in sudden infant death syndrome. And I think one of the ways it could be causing it is by suppressing that enzyme so that the liver can't fix the problem when the sugar gets too low. And then the child goes into a coma.
Interesting. Something that I read in your book, which I was surprised to read actually, is that you said that LDL is a marker for cardiovascular disease. That if it's high, it's a marker for cardiovascular disease. Now, I don't know if you've changed your mind on this, but from what I've read, I've seen that no, it's not about LDL at all, actually. It's about having a low HDL and, as a risk factor, and having high triglycerides and having a high HSCRP and so on and so forth. So, I mean, many people in the health community make a case for the fact that no, LDL is unrelated to cardiovascular disease. What do you think about this? Because you seem to be on the, high LDL might cause heart disease,
Yeah, I mean, I think I say that because everyone believes that that's the case, but in fact, it's actually a very complicated story, but also very, very interesting. And it has to do with cholesterol sulfate, I think, a lot. And so in my book, I talk about glyphosate messing up the sulfation pathways. And so that means that cholesterol sulfate becomes hard to make. And cholesterol sulfate is a beautiful molecule because when you put the sulfate onto the cholesterol, it can go into the blood without having to be packaged up inside an LDL particle. It can just go into the blood. And actually what it'll do is it'll go into the membrane of particles, including red blood cells, but also going into the membrane of the LDL particles. So the liver makes LDL, and the liver depends upon cholesterol sulfate in the membrane of the LDL particle to protect it from damage, from both glycation damage and oxidation damage. And so it keeps what's in the contents inside, it keeps them safe from being destroyed by these destroying agents, which is sugar and oxygen. And so the cholesterol sulfate makes gelled water around the outside of the particle and exclusion zone water, which protects it from the attack from these agents that are floating around in the blood. So cholesterol sulfate becomes a really valuable molecule for making the transport of LDL safe, keeping the LDL safe. And so if you don't have enough cholesterol sulfate, which you don't because glyphosate disrupts the whole sulfation system, and I write a lot about that in my book, the LDL particle is vulnerable to attack by sugar and by oxygen. And so the contents get oxidized and get in trouble. So the oxidized LDL, and it's actually the small, dense, oxidized LDL that's the problem. So after the LDL has kind of delivered its goods, it shrinks. And now it becomes very vulnerable to attack, and it becomes a mess. I mean, all of its molecules inside it are very toxic. And then what happens is it actually gets ushered into and it's fascinating that it gets put into the blood vessels that supply the heart. And the macrophages, take it up, they go in there and they hang out in those blood vessels, creating this kind of plaque, that is so famous as sort of blocking the blood vessels and disrupting. And I think it goes there because it needs to supply the heart with those nutrients. It's looking for I think the heart has a deficiency in both cholesterol and sulfate in the context of chronic glyphosate exposure. And so that cholesterol that's being piled up into the arteries of the heart is waiting to become cholesterol sulfate, but it can't because there's not enough sulfate. And of course, the sulfation pathways are messed up too. And so whenever, in fact, so high homocysteine is a much better marker of heart disease than high cholesterol. But whenever they try to find a drug that lowers homocysteine, it doesn't work. It doesn't make things better. So they don't advertise high homocysteine because they don't have a treatment for it. But high homocysteine is an indicator of sulfate deficiency because homocysteine can become sulfate under oxidizing conditions. So now you have this blood vessel where the immune cells are actually trying to make sulfate out of the homocysteine and trying to attach it to the cholesterol, but glyphosate's messing up all of that. So they're struggling to do that. And if they can, then they can send cholesterol sulfate directly to the heart, which it loves. The heart desperately needs cholesterol sulfate. So it's a way for both cholesterol and sulfate to get transported safely. Sulfate gels the blood, so you can't just put free sulfate in the blood or else you'll have jello and the blood won't circulate. So you have to have these carrier molecules that attach to the sulfate. So those molecules are typically not water soluble, but when you give them a sulfate, they become water soluble and the sulfate becomes safe. So it's a real trick the body uses with all kinds of important molecules. They get sulfated when they're shipped around. And that sulfation pathway is disrupted by glyphosate. So we have trouble with all these different molecules that normally would get into circulation through this mechanism of adding sulfate and then also delivering the sulfate to wherever they go. And so all of that's disrupted by glyphosate. So we have systemic sulfate deficiency and systemic deficiency in all these molecules that are carrier molecules for sulfate at the same time.
That's extremely interesting. Now, since you said cholesterol sulfate, and I started thinking about sunlight, and so I started
Mm-hmm.
thinking about the vitamin D. And a
Mm-hmm.
question I've gotten a lot is because I always advocate for sunlight for vitamin D
Mm-hmm.
instead of supplementation. I intuitively know that
Totally agree.
supplementation comes with problems, and I was very interested to see in your book that you showed studies that show that supplementing vitamin D isn't very effective, and that getting it from
Right.
sunlight, that's the way you're supposed to do it. Um,
Yes.
so what's the problem with supplementation? If you could if you could delve into that.
Yeah, that's quite interesting. So I actually believe that vitamin D is like a hormone, and a hormone is like a signaling molecule that can kind of send messages to cells about the systemic level of situation in terms of some critical thing. And so vitamin D is very interesting because when you get sunlight exposure, the skin cells release vitamin D sulfate, and they also release cholesterol sulfate, both of those. So the skin stimulates the release of cholesterol sulfate from the skin. There's lots of cholesterol sulfate in the skin normally, and the sunlight triggers the release. And the cholesterol sulfate and the vitamin D sulfate both circulate through the blood. The vitamin D sulfate goes to the liver, and the liver takes the sulfate off and then oxidize, and then adds a hydroxyl to the vitamin D using a cytochrome P450 enzyme. And then it becomes hydroxy vitamin D. There's sort of 125 hydroxy vitamin D, which is the active form. The liver does one of those, and then the kidneys does the other one. And now you have the active. So there's a lot of steps that go on with vitamin D. It comes from the skin, it's sulfated. The liver picks up the sulfate, which of course it can use very nicely for something else. And the liver, so the liver is testing both the ability to transfer sulfate or to take sulfate off. It's testing the ability for the CYP enzymes to work. It's testing all these very important enzymes in the liver with the vitamin D to see if they work. And then the vitamin D, the one hydroxy vitamin D goes to the kidneys and becomes 125 hydroxy vitamin D, and then it's activated, and then it can do its signaling that tells the whole world in the body, hey guys, things are good. We got CYP enzymes working in the liver, we got them working in the kidneys, we got the sulfation, taking off sulfate is working too, so things are good. Let's be happy and do good things. So once vitamin D doesn't jump through all those hoops because all those enzymes are being messed up by glyphosate, then you're not going to solve the problem by taking more vitamin D. All you're going to do is overload the liver with vitamin D. It has to do something with that vitamin D, and in so doing, it's distracting it from other things that are probably more important, because the vitamin D is supposed to be just a small amount that signals and says, oh yeah, everything's good. But what you do instead, you load yourself up with vitamin D, not sulfated vitamin D, and it's giving a false signal, because if you get enough of it, it'll probably push through a little that'll tell you, oh yeah, things are good, but really it's not good. It's just you've got tons of vitamin D and only a little bit of it got through, but your body doesn't expect that you got so much, so it's a false signal that says, hey guys, everything's good, but it's not good. And then your body misbehaves, because your cells are operating under the assumption, false assumption of health. And so they're going to behave recklessly in that circumstance.
Wow. Well, now those people got the answer. I'll tell you that. Another thing that I found interesting about your book is you talk about polyphenols and flavonoids. And, in the health community, especially, the part of the health community that really advocates for animal-based diets, they tend to say that, well, no, no, those are not beneficial. Get the sulfate in, get the meat in, and that will produce glutathione, and that's the only antioxidant you need really. And they also say that you've got a lot of anti-nutrients in vegetables and some fruits too. But I mean, I'm guessing that from the papers you've read, you're seeing benefits related to these compounds. So if you could please enlighten these individuals.
Yes, well, this again gets back to the deuterium problem, because these things are ring molecules. There's a whole bunch of different ring molecules in biology. Rings, like a phenol ring is a circle of carbons that go around in a circle. There's a whole bunch of ring molecules that have very, very interesting effects, and all these polyphenols and what are they? What did you say? Polyphenols and
Flavonoids.
flavonoids, right? Flavonoids. So flavonoids are a very good example, because flavin comes from riboflavin, the B vitamin riboflavin. Makes flavin, and then flavin becomes flavin adenine dinucleotide, FAD, and flavin mononucleotide, FMN. FMN and FAD are very, very important so the flavoproteins can kind of pretend to be, they are flavin, so they can substitute sort of for the flavin in those reactions that need the FAD. They're sort of a substitution for riboflavin, you could say. And there's all these different enzymes that use these flavin molecules in a very interesting biophysical way to actually transport protons from the substrate to the product. So these enzymes, in fact, the cytochrome P450 enzymes are an example of that. And those are wrecked by glyphosate, by the way, the CYP enzymes in the liver. That's a huge problem that glyphosate causes. But those enzymes, they bind to NAD, nicotinamide adenine dinucleotide, and they bind to FAD, flavin adenine dinucleotide. And the FAD actually can grab two protons from the source and hang on to them, and then deliver them to the product. And in so doing, there's this whole interesting mechanism called proton tunneling that takes place with those flavins. They have this extremely good skill for moving the protons over from the source to the product. It's sort of complicated biophysics, but they need those flavins in order to do that. And so I think whenever you're taking foods that have a source of flavins, you're helping out that whole system, which is producing deuterium depleted products. It's pulling protons, but it won't let deuterons get through, because it needs this tunneling effect that the deuterons are really bad at, because they're big and bulky. They can't do the tunneling through this sort of narrow space. So it's a trick that the biology uses to not let deuterium come off of the source, keep it behind. Only choose sources that have protons there. I was going to say proteins. And then make sure the product has protons instead of deuterons. And so that's providing a low deuterium nutrient to the mitochondria.
Well, then, what's left is to say, where can people find you, Stephanie?
I have a webpage, stephanieseneff.net. I'm not too active on that webpage. I've been spending most of my time trying to write papers and get them published, which is a very difficult task, because my ideas are so radical that usually there's nobody prepared to review it or to accept it. So I'm struggling to try to figure out how to tell the stories in such a way that a person who's very knowledgeable in biology will actually believe what I'm saying. So I want to continue to publish for as long as I can stay at MIT, because I have an affiliation that will help me. So that's where I focus most of my energy, is in writing papers. And I'm having a lot of fun trying to articulate my ideas around deuterium and package them up into individual papers that tell some part of the story, because the story is so huge. I'd like to write a book, actually, on deuterium, and I will do that, I hope, if I live long enough. And if I ever retire from MIT, I will focus on writing a book on deuterium. I think that's sort of my long-term goals. But I do have a webpage. It has some interesting stuff on there, but I'm not very active it's not currently very active. StephanieSeneff.net. And then, of course, I have, you can contact me through my email, Seneff at csail.mit.edu. And I'm a member of a little group that we call ourselves the New MDs, MDS, which is Moms, Doctors, and Scientists. And that's a collaboration with me and Zen Honeycutt, the one I mentioned, who's doing the measuring glyphosate in all these different food samples, and Michelle Perro, who's a pediatrician. So Zen is the mother, Michelle is the pediatrician, and I'm the scientist. But of course, all three of us are actually mothers. But
I will tell the viewer that if you found this interesting, check out Stephanie's book because
yes,
it's
and I have it here. I can hold it up.
I mean, my God, it's so fun to read if you like
Thank
health.
you.
So
Yeah,
definitely, yeah.
if you like biology, biology is to me a wonderful mystery, and I just love it. And I'm trying so hard to understand every piece of biology, and it's so challenging. But I feel like this deuterium thing, as you can see, I'm very excited about deuterium because I think a lot of things that didn't make sense to me before, as far as the complexities that go into what biology does, it felt like, why does it have to be so complicated? And when you realize that it has to avoid deuterium, all of a sudden a lot of things make sense. So I'm getting much better understanding of why things happen the way they do, as long as I just put deuterium into the mix, and then I can understand what's going on.
Well, I'll be waiting for your deuterium book, Stephanie.
Thank you.
Thank you.